Well, I welcome everyone here. This is our second research roundtable, and I think we're going to have quite a few more are planned. Krish plans quite a few more, so let's get started with this one. I'd like to first point out John Webb, who is our sponsor, and if you want to say a few words.
Thank you, Dave, and good morning, good afternoon, good evening, as the case may be. Got a wonderful international group with us once again today for yet another live and Zoom meeting. They've become pretty popular, apparently, and I guess we're doing something right with all this. I am very, very happy to be able to sponsor these along with MR Resources and Q1 Americas. Of course, MR Resources has been around for 35 years now, supplying very affordably priced, reconditioned Brooker and variant NMR systems, services, spare parts, quench recovery, and the like. And I'd like to ask Krish to say a couple of words on upcoming meetings. Krish, if you would, please.
Thank you, John. Welcome, everyone. And I just wanted to thank today's speaker panels. In addition, I also want to announce the next one that is coming down the pike. The next research topic panel discussion is on October 22nd, that is a Thursday. It will be on effective extraction and application of one bond and long-range heteronuclear coupling constants. The panel will be led by Professor Thomas Williamson at University of North Carolina, Wilmington. And the panel members would be Roberto Gil, Carnegie Mellon University, Alexei Buich at Merck in Kenilworth, and Tio Parella from University of Barcelona. I encourage you to look out for, mark your calendar and look out for email to register for that. With that, I think welcome, and I'll pass it to Dan Iverson for SpinSight update.
Dan, give us an update, please, on Ivan SpinSights.
Certainly. The Ivan team sponsors an NMR forum on Slack called Ivan SpinSights. It's a place where NMR people can discuss anything related to NMR. Membership is open to anyone, not just variant adjuvant users. And you can join the 241 current members with a link I'll send in a chat message. Back to you, John.
Dan, my hat's off to you that you've done a remarkable job in a very short time with kind of revitalizing the whole SpinSights. It never went away, of course, but it was kind of dropped in your lap, and we certainly appreciate it. and Krish, just a little bit further remark on future meetings that I think I heard you say a little bit earlier that we've got speakers sort of booked, if you will, all the way out into February for these meetings. So
that is correct, John.
Got a lot of stuff coming up. And with that, I'll ask Eric to just play a very brief and informative video on Q1, and we'll get started right after that.
We invite you to get to know Q1. Q1 designs and manufactures NMR spectrometers from 400 to 600 megahertz with features for routine use in the research laboratory. Want to upgrade an older system without blowing your budget? Q1 can retrofit AS and UltraShield magnets with complete upgrades, including automation, for less than you think. Q1 offers NMR instruments with excellent performance at an unbeatable price. Experts know the key to performance is the probe, and Q1 offers smart tune and match probes made by Q1 Tech. Q1 Tech has decades of experience and leads the market in innovation. Q1 STM probes have a patented hybrid tuning mechanism, which means faster tuning for improved throughput and unmatched reliability. Q1 offers their 400 megahertz instrument with STM probes in 3 configurations. A 2-channel probe with fluorine tuning on both X and H channels for maximum flexibility. Or fluorine detection can be isolated to the X or H channel to maximize H sensitivity or to maximize carbon and phosphorus sensitivity. Q1 Tech STM probes are also integrated into Topspin and VNMRJ for improved performance and throughput for existing installations.
Thank you, Eric. And I'll turn things over to Dave. And Dave, if you could get things started for us, please.
Okay, let's get going. And before I introduce Emmanuel, who will take over the meeting, I should mention that if you've not been to these meetings before, roughly for the first half an hour to about 12:40, the panel will be giving presentations and discussing their presentations between themselves. And during that time, the chat will be open for questions, and the group will be monitoring the chat as well, and trying to take some of their questions into the discussion. After about 12:40, when they're done, then we open up the discussion to everyone, and we can go as long as we like in talking about the subject. During the talks themselves, I would appreciate it if everyone muted themselves and also muted their video or closed their video. That way we can focus on the pictures of the panelists themselves. So with no more comments, I'll introduce Emmanuel, if you want to take over.
Sure. Thank you. So hi everyone, I'm Emmanuel Hatzakis, and I will open the workshop. Let me start sharing my screen. I hope that everybody can see my screen now. So I would like to thank the organizers for the invitation. In this workshop, we're going to talk about NMR-based metabolomics. I will first introduce you the concept generally, and then Dr. Kostidis will talk about certain techniques, tools, and applications. And then Ms. Fenfen Tang and Ms. Kathryn Williamson will do a demo on how to do NMR metabolomics analysis in real data. So the first thing is, what is metabolomics? Metabolomics is the newest member of the omics technologies. You have probably heard about genomics, transcriptomics, and proteomics. And although there are different definitions in literature about what metabolomics is, we can say that metabolomics, it's simply the science that combines analytical chemistry with statistics and informatics, machine learning, to identify and characterize the small molecules in a system, like a cell, tissue, or organism, generally a biological system. An important thing in metabolomics is that the system is studied or can be studied as a whole. So we have a holistic analysis. We don't really have to focus only on a certain compound. In metabolomics, we often deal with very large data sets, many NMR spectra, each one of many peaks. So what we're trying to do is to extract the maximum useful information from such a large data set. And we're doing this by reducing the dimensionality. This means that we go, as we said, from a system of many spectra and many peaks to a very simple system like a two-dimensional picture, which we can easily work with. In metabolomics, we care about metabolites. So what is a metabolite? Metabolite is any organic molecule which is produced during metabolism and is detectable in a living organism. We're talking about small organic molecules with a molecular weight lower than 1500 Dalton. Most of the time, it's much lower than that. And it includes peptides, sugars, lipids, amino acids, steroids, all of these type of small molecules. Now, there are many metabolites in nature. You can see this pyramid here showing how many metabolites you can find in plants, in microorganisms, in mammals. And that's actually one of the biggest challenges in metabolomics. How you can do a simultaneous and rigorous analysis of all of these different compounds, which have different structures and different properties. The other thing is that these compounds, not only there are a lot, it's not only that we have a big number of compounds, but they're also very diverse. They are produced through different metabolic pathways. Metabolism is a very complicated process anyway, so this makes the analysis even more challenging. You can imagine that you have a certain compound which can be produced by more than one metabolic pathways, okay? So this diversity and these complications make things even more challenging. Now, a big advantage of metabolomics is that it's more sensitive compared to other omics. For example, if you eat something, you don't really expect any changes in your DNA, okay? But you expect a lot of changes in your metabolome, in the small molecules. So this is a really good thing because this is one reason that metabolomics is so valuable. And this is actually one of the reasons that NMR is such a great technique for metabolomics. Because biological systems are so sensitive to changes, we need analytical methods with higher reproducibility. And NMR is such a technique, which is very reproducible, at least compared to mass spectrometry, which is the other main platform we use in metabolomics. Now, in metabolomics, we generally need to answer three questions. Is there any difference between samples? What is the difference between samples? And what is the reason of the difference? So it can be informative, predictive, or comparative. Informative, that in terms they will give us a readout of what's going on to our system. It's often predictive because we use it to predict new samples. Let's say you're trying to find a biomarker to see if a person will develop a disease, for example. Or it can be comparative. You can compare two different groups, okay, to different foods, to different populations, somebody with diabetes and a healthy one, all of this type of comparisons. For example, here the question is, can we use metabolomics, NMR metabolomics, to classify olive fruits according to the variety? So we ran the NMR spectra of olive extracts, and then we did statistical analysis using this NMR data. And we saw that indeed there is a clustering, okay? Now, the clustering depends on the analytical method you are going to use. You can have a whole lecture about the statistical methods you use, more than one lecture actually, about the statistical methods you use in metabolomics, but we can classify in two main categories. One is the unsupervised analysis, and the other is supervised analysis. In unsupervised analysis, the class membership is not known. So the software, your tool, your computer, does not know the group of a sample. So it's totally unbiased. And the main method we use for this type of analysis is PCA, principal component analysis. It's not the only method, but it's one of the main methods. And then we have supervised analysis. So we give our tool, our statistical tool, the information about the samples. So for example, here we said that, look, these samples belong to Sevillano variety, okay? And then the software or the program you have is trying to do the separation. Supervised analysis is generally more powerful than unsupervised. As you can see here, the clustering is better, right? However, there are some issues with supervised analysis that you need to be careful, and we can discuss this if people have questions about that. Here you can see a general NMR-based metabolomics workflow. So we start with a sample of animal origin or plant origin or bacteria or whatever you have. We generally need to do an extraction, especially when we go with liquid NMR. If you don't want to do an extraction, you need to use HRMAS. And actually, that's probably the only way you can do it. You can even do mass spectrometry or something like that if you want to examine the sample as it is. However, most of the time, we are doing an extraction. So the type of solvent you are going to use depends on the compounds of interest. If you care for polar compounds, you will use a polar solvent. If you care for non-polar compounds like lipids, for example, you're going to use a non-polar solvent. Anyway, you do your extraction, you have an extract, you analyze it in your NMR using 1D. Sometimes you can also 2D experiments, but most of the time it's typical 1D experiments. And then you're doing your statistical analysis and you're finding some type of clustering or classification, and also the reasons of the clustering, means which compounds are really driving this clustering. So that's a general NMR workflow. Now, there are two routes for metabolomics in general. One is the targeted analysis, where we need to do quantification of the compounds. We work with concentrations, and it's the untargeted analysis that we don't need to quantify compounds. We're doing spectral comparisons. Very often, although it's not ideal, we don't even do compound identification. Again, we just run a series of spectra and we compare them using chemometrics techniques, okay? In this untargeted thing, we need to make things as identical as possible, because when we introduce extra variants to the systems, it can cause a problem, okay? Here you can see some more information for the target. So for the targeted approach, the biomarkers are often predefined. We usually work with about 1 to 20 metabolites. We can go higher. You can do work now, but now there are tools that you can do work even with 40 metabolites or 50, but this is a general number. Our goal is to identify and quantify the chemical compounds of interest. And again, the statistics you're going to do it using concentrations, okay? The identification of compounds, which is essential here. You can do it with model compounds, with databases, to the NMR, and you can use mass spectrometry. And for targeted metabolomics, we need to have a hypothesis, okay? We need to hypothesize that, okay, I'm expecting these compounds to change. In the untargeted approach, metabolites are not quantified. As I said, often they're not even identified. And the data are statistically analyzed to find patterns, okay? It's spectral comparison, and we're trying to find patterns that can differentiate samples. We're applying chemometrics like PCA and OPLSDA and many other methods. Generally, we need to have a large number of samples. Otherwise, we will have issues in clustering. We will not have really rigorous analysis. Samples must be prepared under identical conditions, and also the NMR acquisition and processing should be the same. So same P1 value, same D1 values or delay, delay time, same phase correction and baseline correction, same zero filling or line broadening parameters. Everything should be the same. And also the samples must be randomized to avoid batch effects. So you want to have, for example, a classification between healthy people and people with diabetes. You don't want to have a classification based on the samples you made in the morning and the samples you made in the afternoon, okay? And there are ways to do this, and you will see it later from Dr. Kostidis. Now, for untargeted analysis, although a hypothesis is not required, you can start analyzing your spectra even without having a hypothesis. However, a rigorous study design is still essential. So, how many biological replicates you're going to have? Are they representative of the population? Did you randomize the data? As we said, all of these things should be considered, although it's untargeted, okay? And for both approaches, compound identification is essential. You can see in many untargeted studies that that's not always the case, but at least we should do our best to identify the compounds which are driving any type of classification and clustering we're going to see. Now, if we have a spectrum like that, the NMR assignment is very, very easy. No one really worries about that. However, in metabolomics, when we're working with biological samples, the spectra look generally like that, okay? So you can see here the NMR spectrum of a feces extra. So you can see hundreds of peaks, many metabolites. The NMR assignment is really difficult here. You can probably do some general assignment like, okay, here are amino acids, here are the sugars, here are some aliphatic or even lipid compounds, but to go to specific metabolites, it can be very challenging. So what type of tools do we have to do metabolite identification? One good tool is NMR databases. There are some databases like the human metabolome databases, which they have spectra of pure compounds, which you can compare with your spectra. That does not work all the time, but it can be very helpful. You can run other type of 1D spectra. For example, you work with phospholipids. You can also run the phosphorus NMR to see what you can get from there. You can do 2D NMR for quantification. And also, if you have commercially available standards, you can do simply spiking experiments to help yourself finding the compound of interest. Now, this is very important because what you generally get from a metabolomic study is a scores plot. You can see it here, which is going to give you the clustering between the two groups, and also the loadings plot, which is going to tell you which NMR peaks are driving the separation. So, for example, you can say that one peak that is important for this separation is the peak at 2.425. So you need to find which compound this peak belongs. So this is what I have at this point. I will stop sharing and I will shift to Dr. Kostidis, who can continue. So Sarantos, you can probably start. Let me stop sharing.
Thank you, Emmanuel. Great. So I'm Sarantos Kostidis. I work in the Center for Proteomics and Metabolomics in the Netherlands and in LUMC, Leiden University Medical Center. And I will continue right away where Emmanuel stopped. He was working about the two categories to generate NMR data for metabolomics, that is using untargeted analysis and targeted analysis. So no need to say more about this. And he also told you that in general, you get really complex spectra. So that is the proton spectrum of urine. And of course, you never have just one spectrum, you acquire more spectrum. And the more you acquire, the more complexity increases. So in order to get meaningful output of your data, you need to go through some steps of pre-processing your data. And I'm going to tell you a little bit briefly about all these steps here, like peak alignment, binning, normalization, batch correction if you have multiple batches, and detection and removal or not removal of outliers. So I will tell you about this in the following slides. Start with alignment. This is, I have an example here of urine NMR, and in this case, on top you see the overlapping because of chemical shifting of creatine and creatinine singlets. Unfortunately, urine has this problem because it has variations in ionic strength or pH, and these variations can cause this chemical shifting. So sometimes we need to normalize urine to the housekeeping compound of creatinine, and you can only do that if you align this. Of course, you cannot do it here, but after alignment, you can separate the two singlets and integrate them separately. And for this alignment, I use a tool called IcoShift, but there are also other ones. And below, I'm also showing an example of how I aligned the triplet of taurine in a more crowded area. Now, to an even more complex example, in the cases that you have multi-cohort or multi-batch studies, like in this one, I borrowed from the literature from Karaman paper in General Proteome Research. They have approximately 8,000 NMR spectra of serum collected in three different sites and in multiple phases and batches. So when they plotted all the 8,000 spectra together, you can clearly see that there is some alignment problem of the peaks, although this is serum. And that's also represented in the medium spectrum below. Then they did RSPA, which stands for recursive segment-wise peak alignment. And they aligned the spectra, and then you also see that then the medium, in the media spectra below, that the peaks of the NMR are higher because of the alignment. And next is binning, which already Emmanuel mentioned that you make a data reduction. This has some benefits that, of course, you reduce the variables that you have to deal with in the final table. And also you overcome a little bit the chemical shifting problems. But of course, the disadvantage is that you lose resolution. Like here, for example, this is a full resolution overlay of several spectra. And here, the bin spectra, where it's been represented by the area of the peaks. In case you want to work with full resolution, there is also an alternative, which I find very interesting. It's that you can work with intervals. So intervals, again, you separate your spectrum in segments, but this time you don't make a binning. Like if I was binning here, for example, this area, I would have 4 bins of 0.02 ppm, and each one of these would have just one value of area or intensity of the peaks in that segment. But working with full resolution, it means that I retain all the data points that I have in my spectrum, and I can analyze these intervals separately, applying typical chemometric methods. For example, I can apply this to PCA, to PLS, to IPL, to PLSDA and ECVA, and that's the letter I here because of interval-based chemometrics. The way this works is very interesting because you can actually apply to the full spectrum or to selected intervals, and then you can get a plot like this, which will tell you a number of misclassifications of your models for each interval. And in this case, we can see that there are a few areas in the spectrum that give 0 mixed classification, and apparently they make up for a very good model, as it can be seen on top. So it's a very interesting application, and there's this group in Denmark that they develop all these tools. I have some references here. You can visit also their website. They're giving away everything for free. They're MATLAB codes, so if you have MATLAB, you can use their tools for free, and they're doing a really nice job. Next, after binning, obviously you have to normalize the data, so I cannot go very deep into this, of course, but generally speaking, normalization is a row-wise process where you can try to correct your spectra based on dilution differences and or for quantitation of peaks. There are several methods to do that, like you can use the total area under the curve or the total length of the spectrum vector. You can normalize to an internal standard, or you can use probabilistic quotient normalization. And then again, after normalization, you can also apply scaling so that you can remove the noise and enhance the information. And scaling is a column-wise operation in this table. Finally, you have to correct for different batches in case you have big studies, because metabolomic studies can last for months or years in some cases. Take, for example, COVID-19 related studies. We're actually running one here in the Netherlands. We started collecting samples in May. The study still goes on, and to be honest, I have no idea when this ends because no one knows what is happening with this. So we're still collecting samples. We're still measuring every week plasma samples from COVID-19 patients. Then the samples can be collected and stored on different sites, and spectrum may be collected in one or more laboratories. So in any case, there might be a possibility that you have some batch effects. And one really powerful tool to control for this is a simple PCA. I have a very simple case here where you have the normal control group, the disease group, and then there's an outlier. Another nice way to control for this, because you have outliers when you have analytical variation or biological variation for some cases. So analytical variation can be controlled really easily with NMR, and my colleague in LUMC, Dr. Asvin Verhoeven, has developed these nice scripts where he extracts parameters from the NMR spectra, like, for example, the field values or the phase values, or SIM values. And this is actually one of my studies, my old studies, where we see that at spectrum after 250, that was a different batch. Suddenly we had a huge field and phase drifting. And so obviously there was something wrong with the instrument at that point, and we had to deal with it. And that was also reflected in the SIM values. So that's a quite nice way to do quality control and correct for batch effects of your data. And the nice thing is that we are living in times that many automatic workflows are developed, and one of these is developed also by us, and the same guy, as I mentioned here, Dr. Asvin Verhoeven. He developed in NIME environment, he developed Kimball, which stands for a NMR-based integrated metabolomics environment. And what this does, it actually takes time domain data and can generate the analysis, all the steps that I had just shown you, and also the steps that Emmanuel mentioned earlier, and give you final outcomes of plots and everything. So NIME can run as it is. So you can, Kimball, sorry, you can run as it is. You can download it, install it in a virtual machine, in a USB, and use it to generate data, or you can modify it as you like, because you can import R or Python available scripts, or you can find freely available. So it's extremely flexible tool. And briefly, it's considered of nodes, and each node here does an operation. For example, here in the beginning, you're just importing your data or metadata and spectra, and then you do the processing, you do the binning, and then you do all the kind of analysis. For example, you can get out experiment info, like I showed you, for example, 90 degrees pulse or sim values or field values. You can get then the bin sizes, or you can even get mini plots where you can quickly visualize all your data and see whether you have good calibration, good baseline correction, and phase correction. And of course, you can plot all your data. And even better, you can also do the binning, either equidistant binning or variable size binning, like intelligent adaptive binning, as in this case. And all this can be easily visualized and modified. So these are more or less the steps that you have to do in pre-processing for non-targeted analysis. For the targeted metabolomics, as you know, you need to have an internal standard and use methods to quantify. And first, I would like to say, why do you need to do targeted metabolomics? Because if we take, for example, this small region here from 2.7 to 62 ppm, where you could probably have 1 or 2 bins in a targeted analysis. In reality here, you have an overlap of 6 compounds. And if you do a targeted analysis, probably you will miss the information from the small guys. So in this case, the convolution and quantification are essential to learn a bit more for your data. And we are lucky to have really nice databases. I'm not going into detail this. There are much literature, and you can find some here. All of them are freely available, but you also have some commercial ones from Bruker, for example, or from Kinomix. And no matter what you find in the database, I always think that, I mean, you are using NMR, and it's so easy to keep the leftovers from our sample preparation and prepare some pooled samples, run a couple of long 2D experiments during the weekend, so we can verify the IDs that we find in the databases above, and we know what we talk about. So quantification, we take advantage of the quantitative profile of NMR. We just have a need just one internal standard. Sometimes this internal standard is not accurately added, so we can correct it like this, for example, using the heretic method or heretic pool based on the poolcon. And we use this equation, and if we take this as a known signal and the heretic as a known one, then we know the area, the temperature, and the 90-degree pulse, and the number of scans. And using this equation, we can accurately calculate how much exactly we have of the internal standard, and subsequently calculate all the other peaks based on this. Yeah, methods of NMR are really moving forward towards a fully automated workflow. That's desirable. The less we have human intervention, the better. I mean, there is the powerful tool of Kinomix, but it's not automatic in any way. It needs a lot of inspection of the data. It's powerful, but it needs experience. Now there's several tools developed here that are also free, which is really nice, to do automatic quantification or semi-automatic quantification. I have used a couple of those, but not all of them, so I cannot tell you a lot, but this is really promising for future of automatic analysis of complex mixtures. And you can even do it in 2D NMR data, like using the Colmar M semi-automated web server tool. You can even do fitting with Kimball, as I mentioned earlier. It does quantification. It actually delivers around 40 metabolites for urine and another 40 for plasma, and that's an ongoing progress, an ongoing project. We will expand this for sure in the future. And another interesting new application I just found out is that from this group in Denmark, it's called Sigma, and it's signature mapping optimized for the analysis of urine NMR spectra. So you import here in Sigma, you import your data, and they have 3 different categories. They are connected to a database. I think it's HMDB. And the algorithm recognizes all the known signals. Then it recognizes all the peaks that they are there, but they are unknowns. And finally, whatever is not recognized as a peak and it's not known, it's considered as being. But in the end, it integrates everything. So you get a full spectrum codification, although for some integrals, you don't know what you have. But it's a quite nice tool, I think, for the analysis of urine. And finally, I'm working in a university medical center, so I would like to talk about NMR in the clinical setting, because it's a really great platform for high throughput clinical screening. And that's because it is a quantitative, reproducible platform. It is robust, it requires minimal sample preparation, and you can even do identification of unknown compounds. So here, for example, I had 32 aliquots of urine of the same sample, and I recorded 32 urine NMR spectra. And you can see that it looks like one because it's a really, I followed the same conditions and I got like a perfect overlay of the data. Examples and applications of this NMR metabolomics in the clinical settings is lipoprotein analysis in blood, for example. This has been commercialized by several companies like Bruker and Nightingale and Lipid Science. And basically what it does is it uses the NMR of serum or plasma, and it calculates the high LDL or the LDL lipoproteins in general, like HDL, VLDL, and LDL, and so on. And why this is really important, because the typical test you just get at, okay, you have 130 milligrams per DL of LDL. But using the NMR method, you can actually get the composition of cholesterol, the distribution of cholesterol in this. And here you can see that the smaller size of LDL indicates a high risk, and you can get this information with NMR, although with a typical clinical examination, you don't get this. So it's quite nice application. And another thing is we have done here is using urinary metabolomics. We predicted EGFR, which is connected to polycystic kidney disease. We found just 4 markers in urine spectra, which we could use to predict EGFR here. Or in the same setting, we could predict the delayed graft function after kidney transplantation, again using the urine outcome. And just in 10 days, we could find some markers that they showed us that some of the grafts, they will have a problem after 4 weeks of delayed graft function. We could find this in just day 10. And even in the same settings, if we take samples from the kidney grafts, like arterial and venous blood, and do the typical NMR blood analysis, then we also collected needle biopsies, and we did HRMAS NMR. We quantified 40 metabolites here, and the best was that the same samples were later used for histological examination, so they were not destroyed. And we generated a quite large data set altogether, and that resulted in really nice outcome, and especially for the nephrologists and the surgeons in the hospital. Last but not least, NMR is also good in studies with cells. So you can study extracellular metabolites by using the culture medium or the cell extracts. And here we also have developed a nice workflow. It's quite robust, but you use NMR in culture samples or cell extracts. You can actually use mass spec using the same protocol. The difference here is that we just need one internal standard with NMR. And although less sensitive than mass spec, we are still able to cover a large portion of central carbon metabolism. And we have already applied it to several cases here in LUMC. And finally, this cellular metabolomics with NMR can be used also to monitor a stem cell differentiation process. We can actually use chemical inhibitors and or simulate with pro-inflammatory molecules and see what is the product of these enzymatic reactions. Or even more, we can use these differentiated cells, put them all together, develop micro tissues, and then with NMR we have analyzed the metabolic reprogramming, among others. So this I had to say, there are just a few applications that you can do where you can do NMR-based metabolomics. And back to you, Emmanuel.
Sarantos, I'm going to hop in. We had one question in the chat about, is Kimball 2 a time domain analysis? So about for your transformer, how does that work?
So yeah, in principle, as if you take it as it is, then you just give FIDs, it reads the FIDs and it does all the processing like TopSpin does. But like I said, you can actually modify this, and you can skip this. You can give in a subsequent node ready NMR data and do the rest of the analysis.
I see. The question, I think the question was
it
about
works both ways.
So you go from time domain to spectra again, like Fourier transformation, right?
Yes.
Not another type of analysis, like Bayesian or something like that. Okay, good. Thank you. Yeah, thank you, Sharon. That was really great. So many nice applications and tools. So now we're going to have a demo using some more standard tools that people use all the time. So Fenfen and the KIT, we will start with Fenfen, who's going to give us a demo about processing, which I believe it's very, very helpful for people who want to start now incorporating NMR as a tool for metabolomics. So Fenfen, go ahead.
Hi, I'm Fenfen. My apologies due to the technical challenges earlier today. I will just play the video that I have pre-recorded.
Yeah, that's fine.
Here's the small sample data set that I have downloaded previously on TopSpin. I have 5 pomegranate juices and 5 apple juices. Here, I'll give a demonstration on one of the samples, how for basic spectral processing steps are performed. EFP for Fourier transform, APK for automatic phase correction, and ABSN for baseline correction. So when you're done these 3 steps, you will want to check the phase and baseline. Sometimes, depending on the samples or instrument condition, you would want to maybe apply a manual phase correction or manual baseline correction. And you wanted to make sure that all samples are done nicely and consistent, that no extra variance will be introduced to this data set. And the 4th step would be calibrate the chemical shift of all these samples. So the one way that we do it here is to add a standard compound, which is TSP in this case. And you would to calibrate to 0 and do that and carry out that to all the samples. Another way that we do that is by calibrating using the glucose that naturally present in the samples system. That way you can avoid introducing or contaminate the sample or just have one less step. When we are done data pre-processing on TopSpin, we will move to Amix for bucketing or binding. So the first step is to load the sample, the folder first. So this is for the directory of the folder where it's saved. And this is the folder name of the data set. You want to make sure it's all correct so you can successfully upload those data to Mx. And then choose sample 1 to 10. So Mx will overlap the spectra of all the samples. You can zoom in or just to check if they are aligned and matched. So there's no sample were missed out during sample pre-processing. Once you're done that visual examination, you go to Mx, tools and buckets, statistics, a new window will pop up. And then you go to statistics, go to bucket table and go to new to create the bucket table. So our data is 1D NMR and there are different bucket methods. Simple rectangular buckets is the common way, or actually always how we do it. And then you go to these different parameters. So the first is the left or right border to determine the spectral region you wanted to use for data analysis. For the last border, you would usually want to choose a value you can exclude the regions where there's not much peaks or information appearing in the spectrum. And then for 0.5, you would want to exclude the region where the standard compound is added for chemical shift, because though that compound doesn't really provide any information about the sample itself. And then for bucket width, 0.01 or 0.02 is the value we usually use but you can also try a smaller value, a bigger value to play around whichever one will give you the best performance of discrimination of different groups. For integration mode, sum of intensity is what we choose among those, because no quantitative compound was added. The experiment is semi-quantitative, and we would choose scale to total intensity among those different methods for two reasons. One, different samples sometimes naturally just have a various concentration. This is a method to compromise that. Another reason is when you prepare samples, maybe will cause some variations of the sample concentration as well. And this is also for that. And for exclusions, we wanted to exclude, in this sample, we wanted to exclude the water peak. It doesn't really provide much information. So it would be 4.7 to 4.9 ppm. And then next. So now we have completed all the parameters for bucketing, and we would want to create a folder name for this bucket. And next. Mx will ask you to select the samples again. I want to select sample 1 to 10. So over here, this is a chance. If you have samples saved at different folders, you can select yes and choose those samples as well. However, we don't have that, so we'll just choose no. And then statistics, again, bucket table, and you choose open. This is how you would load the bucket from Mx. And the bucket table is loaded and saved at a default directory. You want to go to, in my computer, I have it saved at Mx, and then here. So the bucket table, this would be the, okay. Actually, when you were doing bucketing, this table popped up. So this would be the file. So depending on the platform you use for data analysis, you would choose to change the format a little bit, delimited or tab space or save as CSV. Just make the data table compatible with that platform that you use. So from here, I will hand over to Katie, or if you have any questions, please ask. Otherwise, I'll hand it to Katie for further data analysis on Metabolomics.
Yeah, thank you, Fenfen. That was pretty helpful. So yes, here we saw all the preprocessing using Mx. but the steps are pretty similar with any tool you're going to use. So you have the spectra and you need to do a binning, unless you use the whole thing that Sarantos told before. But for the standard procedure, you have to do a binning, do some normalization, some type of normalization, and exclude the areas which not provide any information but distract the analysis, and you end up with a table. So now we're going to see.
And I think there was a question in the chat too about, is there a way to use variable bin windows in AMIX based on intelligent peak detection, or Fenfen, do you want to address that a little bit?
I'll pass that to Emmanuel because personally I haven't used AMIX for different
Sure.
bins.
Yeah,
We do have yeah.
Yes, so yes, there is such. You can use variable bin sizes. I haven't used it because I should do it, NMR bin, which is R-based, but I think that there is, which is very important if you remember what Sarantos said earlier.
Yes, so there is a way on AMIX. Okay, so now we're going to move to the final kind of steps for this. Once you go into AMIX and are able to bin your data, we need to do some type of statistical analysis in order to see, to do our untargeted or targeted, or I'm sorry, our unsupervised or supervised analysis. I'm going to type in the chat. I'm using something called a tool called Metabolomics. If anyone wants to follow along, they're welcome to. You can just click on that link and it should bring you to this page here. And so this is a pretty user-friendly tool. You just hit start, and then we're going to be doing statistical analysis. So we'll click on that little circle. If you're following along, you can select this option, NMR spectral bins, and hit submit. But what I'm going to do today is show you how we would process the data that Fenfen just got from AMIX. And actually, first let me back up and I will show what the data looks like in Excel before, just so we're familiar with what we're putting into Metabolomics. So this is what our data looks like. We have our rows corresponding to our samples, and as Fenfen said, we have 10 samples, and then our columns are corresponding to the buckets or the bins that we created in AMIX, and that first column is related to where that bucket is on the NMR spectrum. Also, a caveat for using Metabolomics is that we need this second column right here where we put our group identification. So we have two different identifications, apple and pomegranate juice. In this instance, if you have multiple treatments, this is where you would put that. And this is required in Metabolomics. In order for it to analyze data, there need to be at least two groups. If you don't know your group classification, you can just kind of arbitrarily make up the groups to see how your data is clustering. So from that, I will select that I'm uploading spectral bins. You can also upload concentrations or peak intensity tables. And as we said, our samples are in rows and they are unpaired. And then I will choose the file that I just showed you. And when we submit this, the first thing that Metabolomics does, or if you selected one of the practice sets, the first step that it'll do is it'll take you to just a data integrity check to make sure that you have that group classification. And here you can see it's telling me that I have 10 samples and I have 950 spectral bins. If you forget to put that group classification in, it will give you an error message here. So you won't be able to get too far without that. The next step is data filtering, a data filtering option. So like I said, there are some really great resources here where Metabolomics will explain what, it'll give you some literature resources and what your options are for this. Since we don't have over 5,000 features, we are going to do no data filtering on this data. A lot of times in NMR, we'll probably end up doing that. And then we'll hit submit and proceed. Fenfen has already normalized in AMIX, so I'm going to leave this option at none. But it's up to you what normalization you're looking to use. In terms of data transformation, I will log transform the data. Again, this is dependent on your experimental parameters and if you choose to do this in your analysis. And finally, I will select Pareto scaling for this data. And I'll hit normalize and then proceed. And those are the steps. Now we're popped out at choosing the analysis method that we'd like to use. So this is a unsupervised method doing PCA, or we have some supervised methods as well. I'll show the PCA first. For lack of time, I'll probably just show what the PCA looks like today. But we are popped here, and this is our screen of our PCA. So if we click on the 2D scores plot, we can see that we have some separation of, these are our pomegranate samples and our apple samples with our principal components 1 and 2 expressed here. There are some tools for publication or just kind of for messing with this final chart. So you can put your sample numbers on here. You also have the option to make it good for grayscale, or if you click on this little painter, you can choose the shapes that you're using and the color that each sample is represented by. You can also look at the loadings plot. And if you click on the individual points on the loadings, it will tell you what bin that's corresponding to and the differences between the two groups that we're working with. So that's kind of a nice feature too. And yeah, so this is something that's really cool. You can play around with all the different types of analysis that you can do. And I guess I can take any questions if there are specific questions about this, or we can move on to the panel discussion.
Yeah, I think very nice.
I'll stop sharing.
I have a comment, Emmanuel, about this, what Kathryn just showed.
Oh, yeah, go ahead.
Yeah, I mean, what I really like about this tool is not only what you can do, but the fact that as Kathryn was clicking to several tools, you could see on the right side the R script generated. So you, in principle, you can copy all this R workflow and reproduce it in your own R environment, or modify it as you like, or implement some other extra tools. So that makes it very flexible too. And it's not like a black box where you import stuff and generates data, you don't know what happens. So I think that's a cool feature.
Yeah, that's true. And to add on this, it's also a good tool for people who want to start learning about R, at least because they're using R there, to see how the different commands and all of these things. Yeah, I agree. And we have a question.
Yeah, Chris has a question. In the loading plot, what do you look for?
Who wants to say this?
Chris
I can say.
asked. Yeah.
Yeah. So, Chris, yes, in the loadings plot, at least for we use this term especially for PCA, but not only for PCA, but for PCA, the loadings plot, it just gives you all the chemical shifts, okay, which are driving any type of clustering, okay, or classification for a supervised analysis. So, for example, you will see that all the peaks which are far away from the center, from 0, they play an important role, okay? And you can see, for example, that the peak at 2.8, it's really driving the separation between the two groups. And now you need to find what the peak at 2.8 is. And you may find that it's citric acid, for example, okay? Did it answer this question?
I think that's clear, yeah.
So to start a little discussion, there was actually another question that was in the chat earlier that I thought we could have readdressed. So this is kind of about normalization and scaling. Is there a benefit one way or the other between row normalization versus internal normalization? And then also kind of off of that, is there one option for normalization that's better than the other? If we want to have a little discussion about that.
May I comment here?
Yeah, sure. Because that was also, the second part was also my question for you, Sarantos. So yes.
Okay. Okay. So I assume Chris here means when he says internal normalization, I assume he means to normalize to an internal standard. I guess. Right?
Yes, that's the question, yes.
Okay, great. So the problem if you normalize to an internal standard is that you cannot count for dilution effect. So for example, when you collect your urine samples, they are already diluted or concentrated to a different degree. But then you add the internal standard in the same amount. And then if you normalize to this internal standard, you cannot account for the dilution differences. So in that case, you should not do this type of normalization. You can normalize to the internal standard when you want to quantify the other peaks. So when you know the quantity of your internal standard and you want to do quantification of the rest of the peaks in the spectrum, then you can do that. Did I answer the question, Chris?
Yes, thank you.
Okay. No, and I had actually, and I had a similar question, yes.
And with regard to the which normalization method is better, that's a very big discussion. I think it's case specific. Overall, we should before we apply normalization, we should definitely know what exactly it does. So, for example, when you do a total intensity normalization, then you assume that all the intensity in all your samples, in all your spectra, is equal. But then if you take, for example, a case where you have diabetes patients with very, very high glucose in their blood samples, and then you have the control which are healthy, and they don't have that much glucose. This high glucose which will dominate the normalized data in the diabetes patients, and it will reduce the values of the other metabolites, which will not be realistic for the data analysis overall. So in that case, it's not advised to use total area normalization. In general, I find it very difficult to apply this, and usually I prefer the probabilistic quotient normalization because it uses a median sample or a reference sample. It calculates the quotients between this sample and each of the other samples, and then it applies these quotients to its spectrum, and therefore it's even when you use the median, for example, it's not prone to big peaks, confounders in the data. And so I find it preferable to use it for urine, for blood, and so on, over the other methods.
Yeah. And I would like to comment also that although we were typically using normalization to total intensity, and it works pretty fine for most of samples, now in a project that we recently did, total intensity didn't work that good, and PQN worked much better. So, yeah.
You can actually go back to your data and have a look why this, because when you know what it does, actually, then you go back to your raw data and you see maybe some of your samples were, I don't know, they're degraded or they have a huge acetate peak, for example, and that makes total intensity normalization, it screws everything.
And so we had another question in the chat. We have a couple actually. The first one was, does Amyx accept variant data? And is there a free tool to convert variant to Bruker data format? And I think, so Fenfen, you can comment too, but I do not think that Amyx accepts variant data since it is Bruker-based
software, yeah.
However, I will say the reason that we're using Amyx is, among other things, to get the buckets, and there are other tools to do that. So like we said, we just showed Amyx today, but there's an R, I'm sure there are multiple R packages. One that we've used is NMR-bin, and you can pull the data in that way and bucket it in that sense. So I think just using a different tool would be easier than finding a way to convert variant data to Bruker data in order to do that.
At this point, I also want to open this to outsiders to comment on that, because on this particular point that you made, Kathryn, Does the AMIX read the spectrum or does it read the FED? If it is a spectrum, it is just an XY, it's an XY plot.
Yes, yes. So it reads I mean, I don't know if you can feed AMIX the FIDs, but it generally reads you have already done the processing topspin, and then just feed this processed spectrum to AMIX.
Do you think MANOVA is a flexible platform? The one that we use for Magrotech?
Oh, MNOVA, yeah.
Yeah.
I have no experience with this, but I want to comment a little bit. My connection was lost for a few seconds, so maybe you commented already about AMIX and whether it's able to convert variant to Bruker data. Perhaps you can import data to AMIX as a text file. So and then maybe this is possible. But I am sure there are tools, more than one, that converts variant to Bruker format to use them. Yeah, I don't know by heart, but I'm pretty sure I have seen it in the literature that they exist.
Yeah, I remember I have used a freeware some years ago. How do you call it? Spin Sparks, something like that, that it was able to do this type of work. But now we have a comment from Amy, and here that AMIX reads the spectrum, but the search tools all require the data is in Bruker format. Yeah, and Nova will also do binning, yes. Okay.
And they also said MANOVA will also bin data. Yeah. So you can probably try that.
I just wanted to mention here, perhaps Dan Iverson is a better person to answer this, but there is if it is spectrum, it's not a freeware, but there is a tool available to convert a variant spectrum into Bruker format spectrum that will go into a PData node, followed by it can be displayed. In topspin. So, but I don't know if it goes beyond into AMIX and how seamlessly it'll get into AMIX and all that. But I know for sure it can be displayed and processed routinely in topspin once you have a spectrum imported from variant. But it is a command on the variant side. Not in the VNMR side.
Now here you can see from Cell Marcus topspin can read VNMR.
Yeah.
So that's another look. Sarantos, I would like to ask you a question in terms of binning again. So, in your opinion, because we saw recently that a bin size of 0.001 worked better than 0.01, which is a typical value people use. Do you see any drawback of using such a small bin size?
It can be a problem when you have a lot of sifting of peaks, like in the urine case. Then it might be that your model in the end will be a bit too noisy, because in some spectra you will have one bin having the peak of interest, and in another spectrum, some other spectra you will have it in the next one. So if you use too small bins, that's one drawback. And then another one is that it depends also on the sample size and the cohort. So if you're just working with 100 samples and you have like 2,000 or 3,000 or 20,000 variables, then building a model there, it's a bit tricky. Especially if you want to do supervised analysis. So that's also one reason to do data reduction in the end, also to build more valid statistical models. Yeah, but
so
I would like to remind you here one part of the presentation about the interval-based. So that's one really nice, smart way to work with smaller bins or even full resolution data using this kind of methods. Yeah.
So you're saying that too many bins can cause an overfitted model, right?
Yeah, I'm not a statistician. I mean, I'm not an expert in this, but I mean, I want to have a model that I can interpret in the end. And this can be very noisy and difficult to interpret. Yeah.
Without actually interrupting the discussion, I just wanted to make sure that everyone understand that the discussion is open to everyone, so people can join in by audio as well.
Okay. And there is, I think there is also another question about binning. There
are several.
Yes.
Yeah.
So
yeah.
one of the questions that I'm seeing is when you do binning or bucketing, do you need to check that the boundaries are not in the middle of a peak? So maybe getting conceptual about how that works.
Yeah.
To the intelligent being.
Yeah, and actually I saw you a nice feature of Kimball too is that you can visualize very easily all your bins, and you can also do adaptive intelligent binning. So you can fine tune it in the end and make your bins in such a size that they include the peaks. Of course, you will always have overlapping proton. That's a big problem. You cannot avoid it. But more or less adaptive binning is, I think, preferable in many cases. And the good thing that you can visualize it with Kimball and with other software probably too. That's beneficial. With Amix, you cannot do that. That's a tricky one, and you have always to go back and recalculate and everything. You have to do it all over again. And yeah.
We have a question from Jenny and I think Dan. It's kind of similar. So Jenny asks, when you process your spectra, is it better to do that manually or automatically for metabolomics? I think which is similar to, are the results very dependent on the person doing the phasing and the baseline correction?
Yeah, I can comment on this. So you can do it automatically, and I believe that the whole thing is going there. Okay, and all these tools that Sarantos showed before, they include this automatic way, not only for data acquisition, but also for data processing. Now, if you don't have these tools and you have the standard tools, I would say that it's probably better to do it manually because, for example, if you apply baseline correction, like what Fenfen did earlier in topspin, so the same thing is applied to all spectra. But this does not mean that the final outcome will be the same, because this is what you want. You want the same baseline for all of them, otherwise this will distract your analysis. If you have only the standard tools, the ones that we showed here, you should probably do a manual processing, a manual phase correction, manual baseline correction. If you have more advanced tools, like some of the ones that Sarantos showed before, then that's another story. I don't know if Sarantos has any comment on it.
I just want to comment that I mean, in modern NMR spectrometers are very stable, and ideally we should go and aim to generate automatic output, and we should be capable of generating good baseline and good phase. And if we can't, I think we should revisit our experimental parameters in the beginning and check out what is wrong and why we get this outcome. But I agree with you that manual inspection, at least, of all the generated data is critical, especially if you have a lot of data and you don't want to do all the analysis and in the end having something very, I don't know, very strange, and then you have to go back all the way and say, why am I getting this? Yeah, so manual inspection is necessary, at least.
I know this is probably a tangential comment on quite a few people who have listened to my lectures in the past probably expected this. I think the idea of baseline correction and phase correction, the term correction itself comes because we are dealing with frequency domain. There is no phase error fundamentally to correct. And there is the baseline correction is really a few points in the FID that are corrected, which the Fourier transform changes into a multiple points being corrupted in frequency domain. So I think that the group, one of the things that I would challenge this group, I am getting old, it challenges group is to start thinking in terms of approaching any kind of time domain analysis. That's the reason one of my earlier question Kimball 2, that's Kimball 2, I believe. Is it a time domain analysis? Not that it accepts time domain data, but actually a time domain analysis or not. Many of these issues with phase correction and baseline correction and automation, and those efforts can be directed towards potential time domain analysis algorithms that are much there are much better mathematicians than I am available, and those people, I think we can focus on this in this area particularly. That's my comment. No question on that.
Well, just a comment on what you said about Kimball. I mean, I know the developer is watching at the moment, so if he knows something more, he can intervene, of course, in the chat. But like I said, Kimball 2 is just a workflow developed, but the whole environment is easily it can be modified. So if you have some tools, some software tools and algorithms to do a time domain analysis, then probably you can implement it. Of course, you will need to have some bioinformatician knowledge, but yeah, you could do that, I think.
Are there questions from the? Yeah, go ahead, please.
Oh, sorry. I just saw the comment too from David saying, I have not encountered a study of rate or error in metabolomics. Have we, Emmanuel, I'm thinking back to in my master's, I remember reading an article about when I think it was multiple different people did the phasing and the baseline. Do you recall an article like that? I don't know if
I don't
know if there's one related to metabolomics, but
probably Robert Powers from Nebraska had probably, but I'm not seeing our own information, but he had done something like that. Yeah, I remember that there was such a paper that they examined this type of factors like batch effects, different people doing the baseline correction and all of these things. I can, David, if you can hear me, I can look for it.
I think it's an open area. I mean, I think we need something like a Metabocon where somebody posts raw data publicly and we just let the whole world work on it and see what we learn from it.
Yeah. This is done partially for NIH-funded studies. The NIH requires people to put the data in some banks, but not for everything.
Yeah.
Another question I have, if this metabolomics world have tried is multiple vendor-based data, meaning the same studies, if multiple centers are doing similar studies and you want to share data for analysis and they have different vendor spectrometers, how much we need to worry about analyzing them together?
That's a good one.
Yeah. I probably, Krish, this will be one of the projects from Fenfen, Ph.D. Because we were talking last time to do something like that. If you remember, Sarantos showed a slide before that, a drift in the field or in log phase values. So I assume, I don't know right now, but I assume that you expect this type of differences from one spectrometer manufacturer to another. So, but this is something we would like to test with Fenfen.
I think at least they can agree in the beginning of the study that they will follow the same experimental parameters and bulk sequences. Okay, same delays and same everything. Of course, then they might have different probe technologies and so on. So, to be honest, I cannot answer this. I'm not. I don't know.
Me neither. I also.
I mean, I know there are many interlaboratory studies between Bruker machines. Probably there are also between the variant machines and so on. But I'm not aware of something with different vendors.
Yeah.
I think Adolfo posted a link where such a study was done.
So Krish.
Oh, there's someone mentioning that there is a study. Yes.
Right.
Okay. Thank you.
Yeah.
We will check this. I will do that. I have been also been a part of a study, but using concentration quantifications, okay? And I think this is okay in my opinion, but for untargeted analysis, yeah, let's read the paper that Adolfo Botana sent.
Yeah, but in the end, yeah, NMR is quantitative, and if you're able to provide, even for a targeted analysis, if you're able to provide the peak areas and everything, then probably this is feasible, I guess.
It should be. It's just that because the area depends on many things. Like as you said, it's what type of probe you have, what type of RG value was used there. I mean, the area depends on some things, but yeah, it would be nice to check.
This may be a little bit more generic question. In the QNMR world, meaning the small molecule quantification validation methodology, they are coming to good consensus lately of a standardized practice of how to do validated QNMR. I mean, there are validation workshops going around. There are conferences that are around such QNMR methodology, not to mention QNMR Summit that come on a yearly basis. So is there something like that in the metabolomics world where there is a generally accepted procedure or gotchas that you need to watch out? Is there something that happens? I am not familiar with it, but is there something that happens that somebody want to comment on that?
Yeah, maybe I can comment also. So in general, with the parameters we are using, we're trying to increase the delay and acquisition time so that we allow most of the protons to relax sufficiently. And of course, we can never achieve, we need too long times to achieve full relaxation. But then there's this nice alternative from Kinomex, where they have developed all these mathematical models, and you can quantify with the convolution your compounds, and they have accounted for this relaxation error. Or the other thing is that you can actually correct for its metabolite as long as you know that you want NT2 values. And then you can manually correct your quantification in the end. So there are ways, and there are people who are working on this too, because there is one camp that wants a targeted analysis, but metabolomics are also moving a lot towards a mechanistic, towards activity metabolomics and a mechanistic insight. So in that cases, you need quantities and sometimes absolute quantities.
I was going after a different direction in that it's not about the absolute quantity, but I was looking at more, I heard in the beginning of this talk, you were talking about how consistently spectra need to be acquired, processed, and in order to avoid variations and all that. And that consistency pattern of what that consistency is in terms of data acquisition or processing tools, is there a consensus among practicing metabolomics people that is agreed upon by a bigger community?
Yeah, absolutely. There are SOP, standard operating procedures, between labs, and for example, everyone who is analyzing urine or serum, more or less, they're using the same same parameters. So in the end, you can compare your data, and yeah, absolutely. The same goes for cell extraction and everything. And I think there are also many efforts to deposit your metabolomic data in a central environment so that everyone can use it and reproduce it. And so, yes, we're moving to this direction.
Okay.
I see that there is a comment about Kimball and the discussion we had about the time domain data. So this is
I think
your answer.
Sarantos, you are breaking. You are breaking.
Developer. Kimball does Fourier transform, automatic reversing, and so on.
Yeah.
Mm-hmm.
Are there other questions coming from the attendees?
I see here.
Okay, can you hear me now?
I see here.
You're frozen, but
oh, I'm frozen.
no, Sarantos is
sorry.
right
Can you
now,
hear
but we
me
can
now?
hear him. I think. Yeah, we can hear you. We just can't see you.
We have a question here from Morgan. What are some ways you can avoid overfitting when conducting a supervised analysis? So I'm not sure if there are ways to avoid overfitting, but you can check overfitting. So for example, if you use PLSDA or OPLSDA, which are probably the most popular techniques, not the best necessarily, but the most popular, there are some quality indicators like the R-square and Q-square, which shows if your model performs well only for the training data set and not very well data set. You can do permutation tests, you can do CV ANOVA. I mean, the best thing is, as we discussed also with Sarantos, is to have a separate, group of samples that you can use it to see how they fit in the model you generated, okay? Given that you have enough samples to do that. And also, we all should probably try to explore other techniques, like, for example, everybody, at least in our field, is using PLS and PLSDA and OPLSDA. There are the techniques which are not prone to overfitting at least that much, like random forest. So there are other supervised techniques you can do in addition to PLS and OPLSDA.
On that score, Emmanuel, is there a, I don't want to divert this discussion to statistical methods, but is there a big picture difference, or I'm sure there is a difference, benefit of one versus the other between PCA and the PLSDA?
Yeah, there are, and I'm not a statistician either, so I don't know everything, but yes, I mean, first of all, there are two different types of analysis. One is unsupervised.
Yes.
So your computer does not know who is who. It's totally unbiased. The whole variance of your system will be reflected in your PCA. And that's why, in terms of publications, it's really good. Always show your PCA, regardless if you see a good result or not. People will use it as a quality indicator of your data. Now, PLS or OPLS, it's a supervised technique. So you provide this information to your computer. You are saying, okay, look, this spectra belong to this group of samples, and this spectra belong to this group of samples. Try to find a separation. The problem is that your tool, like Metabolomics, we showed before, or Simca, which is a very user-friendly thing, or R, or whatever, since it knows who is who, it will make a separation anyway. So the question is this a real separation, or the model used the noise of the spectra or anything else to do? This is where these quality indicators I mentioned, like R-square or Q-square or permutations, CV ANOVA, can be used to see if your model is good. And of course, the best way is to have an independent set to predict it. But the other thing is, we should try to explore, to get out from our comfort zone, which is PLS, OPLS, PLSDA, OPLSDA, and try other things like random forest, support vector machines. There are other things you can do. You can use other methods. One thing here is that some of these methods are popular because they are good, of course, they have many benefits, but also because the first tools that they were introduced in the field had these tools. For example, if you check the literature, I don't know, 80% or whatever, Sarantos can make a comment on it, but 80% of statistical analysis of metabolomics is done by Simca, a user-friendly software. Great, a good tool, but you know, because it has their PLSDA, people simply use the PLSDA now.
Okay.
Just a comment on this, that I think the best advice is that you always go back to your raw data, whatever you find. I think that's very critical because, for example, we said before about the binning and overlapping and how this can influence your outcome. It might be the case that you get a significant variable that separates the group that is just noise. It can happen, actually. PLSDA can do that. So in the end, I think it's a good idea to go back to your raw data to visualize again your bins, maybe modify them a little bit, visualize your baseline, whether everything is fine. And then you can be sure that, okay, the statistics are good, reality is also good, check, I can go.
Yeah.
We have another question. Are the results very dependent on the sample preparation?
Absolutely.
A lot.
Yeah.
Absolutely. That overlaps a little bit with the question of Chris before. That's why we are following SOPs, standard operating procedures. And also there is this feeling that the less you interact with your samples, like we do with NMR in body fluids, that's a big advantage of NMR over other technologies, that we don't do so much sample preparation. We just take the body fluid, we separate plasma, we add a buffer and an internal standard, and that's it. And it's being measured. Of course, you can do extraction. Of course, you can increase your metabolic coverage in many ways by doing some work in the lab. But yes, even in that case, you will have to follow standard operating procedures. And the literature is very, very rich in this area. There are many people who have done comparative studies, and you can find them in many excellent reviews. What are the best SOPs to follow for the specific matrix you're using? I mean, like Emmanuel mentioned, when you're working the food science, obviously, most of the cases you have to do extractions.
Yeah.
Right. And that's definitely, you will have to have a standard operating procedure, but it doesn't mean that it will work for everything. So then.
Yeah, we randomize during sample prep as well as during running the samples.
Yeah.
This is a very naive question, but now you just brought in food sciences. The question is, pretty much all fruit juices are in water. Why do we need to extract them? Can't we just filter and run spectra as is and see if there's anything that we can find out and avoid any extraction pitfalls? This, again, totally naive question.
I think we don't really do like complex extraction, but sometimes we will do centrifuge to just remove some micromolecules like pectin or something, the solids in there, ruin the spectral for us. Or sometimes the water suppression technique, it can be variance between different samples. So probably we wanted to avoid that. So we will remove the water and then just add deuterated D2O. Yeah.
Yes, I agree. And there are cases that you don't need to do any extraction. Just take, let's say, if you have like the study that you show here, the demo, it was juices, right? You can simply take 300 microliters of the juice, do a centrifuge or filtration, put it there, add your D2O, you're ready to go. Now, if you have, for example, meat, or if you have mushroom, or you have tomato, then you may need to do an extraction, especially if you care about metabolites, which, like semipolar or non-polar, you need to do an extraction in some cases. If you have a tissue, a plant tissue, a fruit tissue.
I see an interesting question from Amy. Anybody want to take that?
Yeah.
Let's see it here.
Have anyone?
Yeah, go ahead, Fenfen.
Have any of you gone on to reuse your metabolomics data on multiple studies or gone back to reevaluate different metabolites that were previously explored? This is the strength of NMR that we discuss, but in practice, is it done?
I have done it for both. Yeah, I have gone to a previous study to see my data and re-measure and reevaluate it with regard to another study. And I have also revisited an old data set because I had made a quantification, for example, with one software, then I discovered another that it does it better. So I wanted to compare, and I re-quantified all the data and compared the two softwares to see which serves me best and what's the best outcome. So yeah, you can do that, and that's a very, very big advantage I think we have with NMR data. I mean, the data is there. You don't delete it. Yeah. You have the FIDs there. You can revisit it after years, and it will be there, and you can do.
I just wanted to sort of get a feel for whether or not people really did this, because again, as in talking to audiences that are more comfortable with mass spec metabolomics, to sort of push the strength of NMR, that's one of the features of it, right? That it doesn't matter what metabolite I'm looking for, the response for all the metabolites is the same. I just was curious to know, people who are actually studying metabolomics and have real samples, do they do this? So thank you.
Welcome.
Yeah, in our lab, I think we have done that also. Maybe the first time they used the sample was to explore the nature of the samples. The focus is the sample, the items. But then when we reuse it again, we already know it's a good model. We will use that sample to test out some new technologies we're developing of NMR. Yeah. Okay.
Are there any other questions from the audience? We are coming to close to 2 hours.
We have one more in here that says, does anybody have an SOP for liver extraction? The one we find ends up with a saturated salt solution.
Oh, so they're complaining also about tuning and matching, right?
Okay. I assume that the one, since they have a saturated salt solution, probably they are using a perchloric acid extraction. That's an assumption I can make, but maybe I'm wrong. Ah, I'm correct. Okay. Yes, I have done liver extraction in the past. To be honest, I don't remember by heart the SOP I followed, but I do have SOPs for most kind of tissues because we're working with them all the time. So for liver, heart, kidney, of course, that I saw you, and yeah, some others. So perhaps I can post it somewhere if I find my SOP or the paper.
On that note, I want to alert everyone that this Ivan meeting, as well as the previous ones, this one will be available in a couple of days as a YouTube video in Ivan website, and all we encourage the presenters to provide that presentations to us, so that will be posted in Ivan. So if you want to refer back, you didn't do a screenshot fast enough, so you can always go back to these videos and presentations. Typically it is about a couple of days or so. Eric is very fast on this. Couple of days he posts them. So please feel free to ask questions to the panelists later directly, or you can always go back to Ivan video and then get your questions answered through that, or maybe it's already discussed. With that note, I think we are coming to close of the meeting.
Sorry, Chris, one more me, because I have a question here.
Of course.
It's probably a little bit important. So could you discuss further the advantages of NMR-based metabolomics versus mass spectrometry techniques? I just want to—
Fantastic.
I would like to make a comment here. So first of all, and of course, Sarantos and Katie and Fenfen can also comment, but so although NMR is not that sensitive compared to mass spectrometry, at least for most of the compounds, because for some compounds you can even have more sensitivity than NMR if their ionization degree is horrible, but most of the time NMR is not as sensitive. That's true. However, it's so reproducible, which is really important for metabolomics. If you see how mass spectrometry people, they make QC samples to correct for, over time changes in the instrument, we don't have this problem. Our sample preparation is generally simpler and much faster, okay? So just would like to make a comment on these questions, and I don't know if Sarantos, Fenfen, and Katie want also to say something.
I always see the two methods as complementary, to be honest. I mean, we all know the strengths of NMR. It is reproducible, it is robust, and so on. It can also give, for example, when you do fluxomics, that's another hot area at the moment together with metabolomics. NMR is also has a key role in this area, together with mass spec. So it is true that now approximately 80%, according to a late review published, more or less 80% of metabolomic studies are done with mass spectrometry at the moment. There's still 20% done with NMR. But everyone agrees that these two methods are complementary. For example, I show you some applications that you can do with cellular metabolomics. This is another very hot area at the moment. And NMR is very powerful to monitor, for example, the culture medium. Because of this reproducibility, you can use it in bioreactors, or you can use it in incubators to monitor the culture medium. And of course, you can employ mass spec later to go deeper into the cellular metabolism and have like 200 or 300 metabolites if you wish. But NMR still has a role, and in my opinion, a very important one.
Yeah.
And we should always see it as a complementary method, not as a competitive one.
Only on the chemometrics point of view, but also from compound identification, okay? So if you find a strong biomarker and you would like to have more on this, so you can find them, then you can have an accurate mass, and then you can use NMR to have stereochemistry. So they help each other in both ways.
Yeah, and of course, also the application in clinical screening that I mentioned earlier. NMR is ideal for this because of this reproducibility. You can use it for years to screen body fluids.
Mm-hmm.
Yeah.
Okay. With that note, I want to ask John if he has any last word to us before I close the meeting.
Thank you, Krish, and thank you to all of our fantastic presenters today. Very, very well done to everyone. We certainly appreciate the effort, and great effort indeed, without question. And certainly thank you to all that spent the last almost couple of hours with us today, and we appreciate it. Look for your next Ivan meeting announcement coming along soon in your email box, and we will continue these for ever and ever.
Thanks, John. I wanted to add something special today I think in my memory, the first time we had two students actually participate in this panel discussion. I think it's an extremely useful, great idea. Idea is by Emmanuel, but it's extremely useful. And I want those of you who are attending, I want to encourage you to think of this as forum for younger generation to come in and participate in this. I think we can definitely give them opportunities to do so. And with that, I congratulate particularly Fenfen and Kathryn for their participation today, and it was extremely well done. And of course, the two main speakers, Sarantos and Emmanuel, but you guys take the second seat compared to these two younger generations. So
I agree with you, Krish. Thank you once again to our students. But not only well done, but very professionally done. Yes. Also. And I think we'd like to stick more on this track, as Krish mentioned. The more the merrier.
Okay.
Thanks for having us.
You're welcome. Thank you, and thank you, everyone. Have a good day.
Thank you.
Hope to see you next time.
Thank you very much. Bye.
Bye-bye.